Welcome to the Explainer. Okay, so have you ever looked at a family photo and just found yourself completely baffled? You see two siblings standing side by side, born to the exact same parents, and yet they look like they belong to entirely different families. I mean, one might be a pale headhead with freckles, while the other is a dark-haired brunette with olive skin. It's a super relatable paradox that makes you secretly wonder if there was, a tiny mix-up at the hospital. Well, today we are going to unravel this biological mystery and discover exactly why you might look absolutely nothing like your brother or sister.
So, to get to the bottom of this, we first need to challenge a very common, very pervasive myth. We're all taught in basic biology that because you get half your genes from your mom and half from your dad, full siblings inherently share exactly half of their genetics with each other. It sounds perfectly logical, right? But nature, as it turns out, is far more chaotic and infinitely more fascinating than just simple division.
Now, what's really interesting about this slide is the hard data we're looking at straight from 23andMe. Notice the full sibling row. Yes, the average percent of DNA shared is 50%, but pay close attention to that expected range right next to it. Full siblings can actually share anywhere from 38% all the way up to 61% of their DNA. That is a massive spread. It means you could technically be far more or far less genetically similar to your sibling than that standard 50% rule implies. Right, so while 50% is the statistical average that we all constantly quote, the reality is this vast, unpredictable genetic spectrum. And to figure out why you might be sitting closer to that 38% mark with a sibling who looks like your total opposite, we are going to investigate the cellular culprit behind this massive genetic variation. It literally all comes down to how our bodies manufacture the cells that make the next generation.
To track down our culprit, we have to draw a hard line between two completely different types of cell division, mitosis and meiosis. Mitosis is basically your body's everyday worker. It takes a single cell and divides it into two identical clones, with the exact same number and kind of chromosomes as the parent cell. This is just for tissue growth, skin repair, and basic bodily maintenance. Meiosis, on the other hand, is nature's ultimate randomizer. It is a highly specialized process designed exclusively for reproduction, and its entire goal is to create genetic diversity.
So meiosis is responsible for producing gametes. Now, rather than carrying a full deck of 46 chromosomes like your standard body cells, these gametes carry exactly half that standard chromosomal deck, just 23 instead of 46. Why? Because when they eventually fuse during fertilization, they combine their halves to create a whole new human with a complete set of 46. But the truly amazing part isn't just the math. It's how the body randomly chooses which genes from your grandparents actually go into these specific cells.
Okay, let's dive into this and actually track the physical movement of these chromosomes in action. A single diploid cell, which just means it has two sets of chromosomes, undergoes not one, but two cycles of cell division. Yet it only replicates its DNA once. The result? Those four unique haploid daughter cells you see at the very end, each containing a single set of chromosomes. But notice how the colors on those chromosomes are mixed up? That is exactly where the magic happens. The very first step in this giant genetic shuffle is a phenomenon called independent assortment. When those chromosome pairs line up to be divided, they move completely randomly to separate poles of the cell. The chromosomes your father inherited from his mother and father don't stay neatly separated. They get entirely shuffled. This means that every single gamete ends up with a completely random mix of the four grandparents' chromosomes. It's exactly like dealing out a fresh, rapid-fire hand from a deck of cards every single time.
But wait, it gets crazier. Let's see how this builds into an even deeper level of randomization called crossing over. This is a highly technical, physical exchange of genetic material. Step 1 is a process called synapsis, which is really just the scientific term for the exact moment homologous chromosomes, one inherited from each parent, pair up intimately, gene by gene, along their entire lengths. Step 2, programmed double-strand breaks, actually sever the DNA. And Step 3, the chromosomes reconnect, literally trading genes with one another.
Now, this incredibly dramatic exchange happens during prophase 1, specifically in what's known as the Pachytene stage, which is just a fun little bit of biology trivia for you. The chromosomes form physical connections called chiasmata. Think of these like incredibly precise molecular zippers. The strands of DNA are broken, swapped, and expertly repaired. Because of this crossing over, the chromatins are no longer identical to the ones the parent was born with. They are brand new, completely recombining chromosomes.
Okay, to make this incredibly tiny molecular process a bit more tangible, let's use a brilliant analogy. Let's imagine a DNA sequence inherited from a mom, one that codes for a specific set of traits, as a simple sentence. The sentence reads, "Imagine a rabbit hiding in the bushes." Every single word represents an allele, or a specific version of a gene.
And this brilliantly illustrates what happens next. The father contributes his own equivalent genetic sentence. It describes the exact same idea, but uses completely different words, or alleles. His sentences conceptualize a hare cloaked in vegetation. Both sentences are complete, both function properly, but the specific genetic vocabulary is remarkably different.
So when crossing over occurs, these sentences are literally cut and pasted together. The recombinant sentence that ends up in the gamete might read, "Conceptualize rabbit cloaked in bushes." This is an entirely new, never-before-seen genetic sequence. You now have a unique combination of alleles that neither the mother nor the father possessed in that exact order. Multiply this by thousands of genes and you really start to see why siblings differ so drastically. All right, let's pivot back to solving our initial real-world mystery, that pale red-haired sibling standing right next to the dark-haired sibling. Once this newly shuffled DNA is locked in, how did the physical traits actually show up? Well, this is governed by dominant and recessive genes. Simply put, some gene instructions are stronger or dominant, and they will completely mask the presence of weaker recessive genes if both happen to be present in the new sequence.
Let's look at the specific genetic switch for hair color. It's known as the MC1R gene, and its main job is to control red pigment in the body. The version of this gene that says not red is dominant. We'll call it a dominant capital R. The version that says red is recessive, a recessive lowercase r. If you have both, the dominant not red wins. You need 2 copies of the recessive lowercase r gene to actually have red hair. And importantly, this gene is a total package deal. The red hair instruction almost always comes bundled with instructions for fair skin and freckles.
So what is the statistical probability of 2 parents without red hair having a red-haired child? Well, if both parents are what we call carriers, meaning they each have one dominant capital R and one recessive lowercase r, each time they have a child, the genetic lottery rolls. There's a 50% chance the child gets a mixed pair, a 25% chance they get 2 dominants, and only a 25% chance that 2 recessive genes will combine to produce that red-haired, fair-skinned, freckled baby. So the crucial point is this. Our mystery is officially solved. The reason our siblings look totally different is all because of those hidden recessive genes. The grandparents passed down the recessive red hair blueprint, meaning the parents were secretly carrying it without showing it themselves because their dominant dark hair genes masked it. When the genetic deck was shuffled during meiosis, one sibling drew the rare double recessive hand, while the other drew the dominant dark hair hand. A single gene out of around 25,000 explains this massive visual difference. Zooming out to the macro level for a second, it is truly worth asking why biology goes through all this trouble. I mean, why utilize this staggeringly complex cellular machinery, meiosis, independent assortment, chromosomal crossing over, and dominant recessive masking? Because genetic variation is an absolute evolutionary superpower. By constantly reshuffling the genetic deck, populations produce individuals with completely unique traits, ensuring that a species can adapt to harsh new environments, survive devastating diseases, and ultimately thrive in a constantly changing world.
We've explored the fascinating, beautiful chaos of cell division and the hidden codes lurking within your own family tree. Every single crossing over event, every random assortment, means that out of all the possible genetic combinations your parents could have produced, you are a literal one of a kind. Which leaves us with one final thought. With thousands of genes shuffling in every single reproductive cell, how many entirely unique, never-before-seen combinations exist right now in your own DNA? Thank you so much for joining me on this explainer. Keep wondering and keep learning.